So thank you so much for having me. It's my pleasure to kick off your Spring 2026 seminar series about some of the work that we've been doing in our lab. And so as you can see here, the title of my talk is Developing a Performance-Based Measure for Monitoring Dementia Risk, Evidence in Alzheimer's Disease. And I know that there's a strong interest among this group for stroke, and I'll touch a little bit about that, about how some of my work, my current work, really stemmed from my prior work in stroke motor recovery. I want to start by acknowledging my amazing collaborators and coworkers who have helped me and helped us develop this line of work over now spanning over more than a decade. And I want to really acknowledge that the charter of Arizona State University, because it has really informed some of the research directions and the goals that we set for ourselves as researchers in our lab, and really making sure that our work has public value and that it serves our local and national communities, and even international in some cases. And so coming back to the title, developing a performance-based test for monitoring dementia risk, there's a couple things that I want to highlight. The first is monitoring dementia risk. And so we know that some assessments, and you're probably very familiar with some of these, there are a number of objective assessments that are used to evaluate individuals' cognition. Those are listed here. Some of you probably use these in either your clinical practice or research programs, and these really help us evaluate levels of global or domain-specific cognition. But a really important part of understanding dementia and dementia risk is not only cognition, but also the level of daily functioning, or how an individual is impaired in their daily functioning, because the definition of dementia is cognitive impairment that is severe enough to affect one's daily functioning. And the ways that clinicians and researchers evaluate daily function are shown here at the top. And some of you may also be familiar with some of these. But what I want to highlight is something that all of these listed measures, and this is not an exhaustive list by any means, but these are some of the most commonly used measures to evaluate daily functioning in the context of dementia and cognitive impairment, is something that these all have in common. And really, they all involve subjective reporting from an informant or a collateral source, which is kind of typically a spouse, an adult child, a close friend, et cetera, somebody that can sort of endorse sort of the level of impairment or level of independence that an individual may have. A really commonly used measure is the clinical dementia rating system. This is used a lot as inclusion and exclusion criteria for prevention trials in Alzheimer's disease. It's also used a lot in characterizing individuals in Alzheimer's disease and MCI studies. But this is just one example of an item on the CDR, and you can sort of see what is being asked of the informant. And this is, as shown, the community affairs questions. Is the person still working? Do you think that memory or thinking problems contributed to this person's decision to retire? And do you think this person has difficulty in their jobs because of cognitive problems? And it's sort of a yes, no, sometimes, I don't really know. And while this can provide a lot of great insight, a lot of these responses are subjective, which is good, but also potentially problematic. And so the way that the CDR, the Clinical Dementia Rating System, is scored nowadays is a CDR sum of boxes, which ranges from a 0 to 18, where higher values indicate more impairment. And what was really interesting in this paper that came out in 2024 was looking at a large data set within a large clinical trial. We actually saw, or the authors saw, higher CDR sum of boxes scores if the informant was female, an adult child, or had more frequent contact with the participant, which kind of makes sense. But I really want to highlight that in one of the sort of groundbreaking prevention trials for the drug lecanemab, the difference between the placebo and intervention arms of this phase 3 trial was within the range of what we saw, this normal variation in informants. So what the difference in the effect of the drug was similar to the effect of the informants on the score, which is interesting in and of itself. Kind of in addition to this, just sort of practically, we know that nearly 25% or possibly even more older adults in the US are what we would call solo agers, meaning they really don't have an informant or at least a reliable informant that can speak to how they're functioning in their daily life. And something I'll touch on later in my talk is how these sort of predefined daily activities that are specified in some of these measures may not be relevant to certain populations or certain individuals. And so I really want to emphasize here that there is a clear need for tools that can objectively monitor the impact of cognition on daily functioning without a reliable informant. I also want to highlight that we are not the first to really explore these. Again, a non-exhaustive table is shown here of some objective or performance-based measures shown on the left, abbreviated on the left. And you can kind of see what they entail and some different features about them. But as you can kind of see, they are either very lengthy, they require formal training, or there is a large sort of equipment burden, which really makes it difficult to implement on a large and systematic scale. So our goal in the lab continues to be to develop a brief, easy to administer, and objective measure of daily functioning, and possibly one that's specific to Alzheimer's disease. And the specificity to Alzheimer's disease is really driven by two needs, both in terms of clinical trials as well as just kind of primary care, is really this need for functional outcomes to help monitor the effects of these drugs in particular trials, as well as for just monitoring patients outside of specialist care. And there's kind of this new term that the Alzheimer's Association has really kind of pushed, and that primary care is now, like it or lump it, the front lines of dementia care and dementia screening. And so understanding the workflow of primary care and the really long wait times that patients may have to go from primary care to specialist care in the context of dementia really warrants the need for something that aligns with our goal. And so what I'll be talking about today really is a performance-based measure that we've developed in the lab. And this is just a video of a person doing the assessment. And the origins of this test really came from my background in stroke rehabilitation. As I mentioned earlier, that's what my background is. My doctoral and postdoctoral training was in the area of stroke motor recovery. And I initially designed a test very similar to this, really as a task-specific training protocol for individuals post-stroke or with post-stroke hemiparesis to practice task-specific activities to promote use-dependent recovery. And really what we found when administering this or having individuals practice this task over and over again is that their level of cognitive impairment, really just evaluated by global measures, affected how much they improved or learned this task. And so that really stemmed this line of work that I'll talk about now. So I want to just revisit this idea that the assessment is brief, performance-based, and objective. So in my lab here at ASU, we have an instrumented version of this where we put sensors or a way for us to track an individual's motion during the task. This is just tracking the movement of the person's hand from a top view and a side view as they perform the task. So we can do some very detailed kinematic analyses. But for the purposes of this talk, I just want to talk about the measures or the ways that we can evaluate performance based just on time, how long it takes them to do a given trial. And so they have to scoop 2 beans at a time with their non-dominant hand, and they have to do 15 reaches per trial. So we just simply time them with a stopwatch. And what we see is that when we had people, older adults specifically, who were cognitively unimpaired, motorically unimpaired, the non-dominant hand was significantly slower on their attempt at this task compared to the dominant hand. So this was something that allowed us to explore this a little bit further. And so what I want to show you here now is task performance from 2 individuals over 6 consecutive trials or attempts at this task. And so on the y-axis is task performance measured simply as time, And you can see that on the x-axis is just the practice trial, so trials 1 through 6. And higher values would be considered worse. That is, they're just moving slower, or they have longer trial times on any given trial. And you can kind of see two different sort of performance levels. An individual who is kind of getting faster over time and has pretty stable performance on the left. And we see an individual with kind of a lot of variability in their trial times as shown on the right. Now, there are a lot of different patterns within an individual or set of individuals' performance across these 6 trials. But really what we've been able to show and what evaluating multiple trials on our task allows us to calculate and evaluate intra-individual variability, or IIV, which is not a term that we've come up with. It's actually a well-established term within the cognitive literature and cognitive aging literature that reflects how stable someone's performance is across trials of any type of assessment. And there's a lot of literature in the field that really shows that high levels of variability are often associated with more cognitive impairment. And so we measure IIV, or this intra-individual variability, simply as the within subject standard deviation, how much variance there is across these 6 trials in trial time. So, so now I want to show you, excuse me, how this intra-individual variability, we have been able to show that it is associated with a number of factors. On this particular slide, I'll show you some data that we've published that is associated with Alzheimer's disease staging and biomarkers. So what you see here is just individual participants, someone who's been diagnosed with mild Alzheimer's disease based on clinical criteria, an individual who's been diagnosed with amnestic MCI, again, based on clinical criteria, and somebody who's been identified as cognitively unimpaired, also based on cognitive criteria. And you can see how not only sort of the average trial time, but also the variability in trial time increases over the course of these 6 trials. This is just individual data, but we've been able to demonstrate that across Alzheimer's disease stages, as shown here, we see an increase in standard deviation, this sort of intra-individual variability, as we increase across disease severity and these Alzheimer's disease stages. We've also been able to demonstrate associations with Alzheimer's pathology. The gold standard for evaluating Alzheimer's disease pathology is PET imaging, positron emission tomography. And again, these are just exemplar cases of 2 individuals who in this case are diagnosed with amnestic MCI. So they're in sort of that preclinical stage where they have not yet met the criteria for dementia due to Alzheimer's disease. But these are 2 individuals, one who would be considered amyloid negative based on their PET scan, and that would be case A. And case B is somebody who would be considered amyloid positive based on their PET scan. And again, these are just individual cases, but in the paper listed below, we've been able to show an association between performance on our test and the level of amyloid as measured through PET. One other biomarker of Alzheimer's disease is hippocampal volume. And really, it's thought that accelerated atrophy within the hippocampus relative to the whole brain is strongly associated with Alzheimer's disease. And here we showed that hippocampal volume is related to our task as when it's measured with intra-individual variability. And we've been able to replicate this hippocampal volume finding in a number of papers as listed here through a number of different analyses. And hippocampal volume was measured just through structural neuroimaging. And something that is sort of hot off the press, my PhD student, Alexu, who was actually on this call, has been looking at some new data that we've been able to acquire with a small internal grant to look at plasma biomarkers and cognitively unimpaired individuals. And the results are showing that performance on our test is related to basically A beta 4240, which is another measure of amyloid, but not some of these general neuroinflammatory plasma biomarkers. So this really suggests some level of specificity of performance on our test and Alzheimer's disease specifically. Moving on, though, to the overall goal of what we're trying to accomplish in the lab is how does this sort of map onto daily functioning? And so when we look at one of the commonly used measures of daily functioning, in this case, it's the Quick Dementia Rating System, or QDRS. The QDRS is as noted in the Q, a quick assessment that evaluates these 10 domains. And what we find is that there's a significant relationship between people's intra-individual variability and their scores, the informant scores on the QDRS. I really want to kind of highlight here, because it's not something that I mentioned early on, but just from a psychometrics perspective, I really want to kind of note the ceiling effect, or sort of that clustering of a lot of scores around 0, or really lack of impairment as measured by the QDRS, that potentially is problematic for measures that are trying to detect early impairment or early decline in the sort of more prevention stages. One of the nice things about the QDRS is that you can also estimate a total or overall clinical dementia rating score. So these are estimated CDRs, which again is kind of thought to be this gold standard for clinical trials, where an individual in a prevention trial would want to have a CDR of 0.5 or higher. We see an association or relationship between the intra-individual variability and CDR. This total CDR is estimated by the Quick Dementia Rating System. So this really sort of validates and has helped us validate our assessment as a measure that can estimate or be a proxy for the level of cognitive impairment's impact on daily functioning. So something that many of you may be thinking is, well, what if the person has motor impairment? This performance-based test involves motor, sort of motor capability. How does your test differentiate, or how does it rule out just whether somebody has, motor cortex damage or essential tremor or weakness? So how do we take that into consideration? And so this is a question that we got a lot and we continue to still get in our grant reviews. And so we actually first tried to look at this using some secondary data or existing data from a cohort of individuals who had done our assessment and were diagnosed with Alzheimer's disease. And then we also looked at a cohort of individuals who did not have any cognitive impairment, but had been diagnosed with Parkinson's disease. And based on their UPDR scores and their Honyard, they were anywhere from mild to moderate PD, but they did have motor impairment. And we considered also as a control group, a group of individuals who had neither an AD nor PD diagnosis, and they would be considered neurologically intact. So really, we have a cognitively impaired group, a motorically impaired group, and then a neither impaired group. And these are just the demographics and clinical scores there. And really what I want to highlight is when we look at the ability of our test to differentiate just based on performance between whether somebody was in the AD group or the PD group, that is cognitively impaired or motorically impaired, we see a very high level of discrimination. But the value of this control group now is when we try and differentiate between two cognitively unimpaired groups, just one that has motor impairment and one that doesn't, our assessment really can't differentiate between those. And so what I want to highlight is the way that we evaluate performance on our test, that is using the intra-individual variability. It appears to be sensitive to cognitive deficits, not sensory motor deficits. And really this suggests that the IIV measure doesn't really evaluate the same constructs as a sensory motor test, such as grip strength, like a grooved pegboard, or maybe even some sensory deficits as indicated here. And so I want to just kind of unpack what I mean by that a little bit more because I think it may be relevant for this group. And I want to do that using some data. So what I have here is, again, trial time, or kind of how long it takes them to do and perform an individual trial for two uh, co cognitively and motorically unimpaired individuals over these 6 trials. You can kind of see those there. Then I'm showing performance on across those 6 trials for an individual who has been diagnosed with Parkinson's disease, and this is when they are on their levodopa, so they're on their Parkinson's medication, and then that's the dark blue line, and then the lighter blue line is also an individual who had was an individual post-stroke, chronic post-stroke. And you can see that on any given trial, their performances take longer. But when we look at how these motorically impaired individuals, the individual with Parkinson's disease and the individual post-stroke, you can see that their average trial times across those 6 trials are much higher than the unimpaired individuals, but their standard deviations, or that IIV, is within the same range as those motorically unimpaired individuals. So keeping in mind that these individuals differ based on their level of motor impairment, but they don't appear to differ based on their level of cognitive impairment. And this is further demonstrated when we look at average trial time versus standard deviation in trial time in correlations with this sort of the groove pegboard test, and we see a differential association between mean and standard deviation trial time with respect to a clinical measure of motor function. And so this hopefully can help kind of disentangle the associations between cognition and motor function, cognitive and motor function, when it comes to what we're measuring with our test. And when we look at other cognitive measures, some of which you may be familiar with, either global cognitive screening measures or very in-depth neuropsychological assessment, something that kind of continues to emerge in the neuropsychological literature is really how participant characteristics or demographics can influence an individual's score on a number of cognitive measures, which has sort of led the field of neuropsychology to really see the importance of normative scoring or normalizing scores based on participant characteristics such as ethnicity, race, sex, or level of education. But in an ongoing grant that we have from the Alzheimer's Association, we really wanted to evaluate, is our test sort of resistant to participant characteristics, particularly ethnicity? And so using equivalence testing, we're starting to show that the task IIV is equivalent between individuals who either identify as being Hispanic Latino or not Hispanic Latino, when you control for the level of diabetes, or when you control for the presence of diabetes. And so it's kind of resistant to or not affected by some of these participant characteristics. And when we look at that in relative to other well-established neuropsychological assessments that are listed or abbreviated here, compared to those tests, the Hopkins Verbal Learning Test, the Symbol Digit Modality Test, which are used to evaluate certain aspects of cognition in neuropsychological assessment, those tests tend to show effects of sex and or education, whereas our performance-based tests do not. And we've been able to replicate this in a larger sample with some other cognitive tests as well. So a quick recap of what I've just thrown at you is that we've developed a performance-based, an objective measure that takes just a few minutes to administer. I've shown you some data that supports its association with Alzheimer's disease staging and biomarkers. We've been able to show that it is associated with existing measures of daily functioning. It appears to be more sensitive to cognitive deficits rather than sensory motor deficits when we look at that intra-individual variability measure. And it is resistant to participant characteristics. And so it seems to be highly generalizable and usable in a lot of contexts. And so going back to the larger needs within both clinical and research fields, to really think about scalability and generalizability, the goal we wanted to pursue further was, can this be collected reliably, remotely and possibly unsupervised? And so to do that, we are partnering with MindCrowd, which is a large electronic cohort based, uh, well, uh, conceptualized by my collaborator, Matt Hunnelman at the Translational Genomics Research Institute and partnering with a number of other institutions nationwide. And at this point, MindCrowd overall has assessed close to 850,000 people nationwide on some brief memory tests and collected a lot of demographic information. And what we wanted to do was to leverage this MindCrowd cohort to see, can we evaluate or have our test administered remotely and unsupervised? So in a subset of individuals in the MindCrowd cohort, we asked them, hey, can we mail you something in the mail, this performance-based test, and can you do it at home, kind of at your kitchen table by yourself? Tell us about how you administered it. Upload your scores, and we'll integrate that back into your MindCrowd profile. So that's what we did. And in this study that is now several years old.
By newspapers, they're calling it an exclusive report. They quoted the chairman of ADC in Kogi State.
Okay, thought that was a question. So in the study that's now several years old, we sent the kit out to 177 people nationwide, and we compared their scores against an in-lab cohort that we collected face to face. And I like to say that these are the most interesting, boring results, or perhaps the most boring, interesting results, because we see equivalence between the in-lab and MindCrowd scores that really illustrate or support the reliability of remote and unsupervised data collection. And we are moving forward with the MindCrowd cohort in part to really further understand the equivalence across ethnicity and how some other demographic factors may or may not influence performance on our test. And the MindCrowd cohort is a great cohort to partner with because about 18% of its of the participants identify as Hispanic Latino. So we are really statistically powered to look at these effects. So there is this overall MindCrowd cohort that we are sending kits out to. But I also want to highlight that a new collaboration or a new project that's starting is leveraging this MindCrowd mobile lab. And really what that is ultra low field scanner, the hyperfine scanner that you may be familiar with, is packed into this trailer and is driven around to rural and otherwise difficult to access places throughout the state of Arizona. And they're able to do some ultra low field neuroimaging, as well as collect blood for plasma and genomics analysis. And this is the start of a recently funded project by NIH in helping us develop a performance-based measure of daily functioning for screening Hispanic, Latino, older adults for Alzheimer's disease risk. And you can kind of see there on the right what the sort of overall demographics of the mobile lab are at this point. And so thinking about this method for monitoring and triaging patients outside of specialist care. So really like thinking about primary care. An important thing is to consider who would be likely to progress over time. Because if somebody is relatively stable, there's not really cause for concern at that particular point in time. But identifying individuals who might be at a higher risk for declining over the next year or two is really important for primary care. And so we went back and looked at some longitudinal data, and we calculated the amount of change in the mini mental status exam to look at the change in cognition, as well as the QDRS to calculate the amount of change over 1 year in daily functioning. And we used Z-scores from a regression-based change formula, where 0 indicates stability. So if an individual has a 1 year change of 0, they were stable, and anything higher than that or lower than that, depending on the scoring of the assessment, would indicate more change or more decline over time. And what we see is that our IIV measure, the standard deviation at baseline, could predict how much a person declined in terms of cognition and daily functioning over 1 year. And this is across Alzheimer's disease stages. The average performance, or that average trial time, did not have predictive value in cognitive or functional change over 1 year. And I know that 1 year is not a very long time frame. And so I was really interested in identifying whether we could kind of predict more or predict decline over a longer time frame. But to do that, I needed to go to Sweden, which is what I did in the spring of 2024 on a Fulbright fellowship. And there I was able to leverage 3 population data sets: the Octo-Twin study, the SATSA study, and the gender study. And the reason that I wanted to go to Sweden, and the reason that that these data sets were of such value, was because in these data sets, there were common data elements, specifically the mini mental status exam, so a cognitive measure. And there was a functional fine motor test, which is shown here. It involved timing individuals and how long it took them to insert a plug into a socket, a key into a lock and turn it, inserting a light bulb into a socket, putting coins into slots, and then dialing the numbers 1 through 9 on a rotary phone. So that's the ADL apparatus shown here. But it also collected grip strength and gait, that is the time to walk 3 meters out and back. And in doing so, I was able to calculate T-scores to allow comparisons across measures in order to say which of these measures perhaps tracked with or could predict the onset of dementia. Also in these data sets, there was clinical consensus on whether an individual developed dementia or not over the course of their involvement in the study. And I was able to then use linear latent growth curve analysis that was centered on either the time of dementia onset or their last study visit, and that would be the intercept. And really, what this allowed us to do is look at very long-term changes in these different motor-based measures or performance-based measures, and which ones did or didn't track with eventual onset of dementia. And although we don't have our particular measure in these studies, I think it doesn't take too much squinting to appreciate that there's probably some similarities or similar constructs that are being tapped into with the ADL apparatus and our assessment. And so really what I want you to appreciate here is that we have 3 different motor tasks or motor-based tasks that we could compare side by side. So that's what I'll do here. On the y-axis is the T-score for that given measure. And then on the x-axis is time up until the time of dementia onset or last visit. So we're looking back in time between a group that did not develop dementia and a group that did eventually develop dementia that was diagnosed at time 0. So this is what the data look like for that gait time, which you can think of as gait speed. This is what it looks like for grip strength. And then this is what it looks like for that functional fine motor test, that ADL apparatus, where gait and grip really can't really discriminate or doesn't really show differences between these 2 groups. not even until the time zero, whereas about 10 years prior to the onset of dementia, or the prior to the diagnosis of dementia, excuse me, we start to see these groups diverge. And the mini mental status exam is just shown in panel D to sort of show, okay, as you would expect, we would see a more rapid decline in cognition in the dementia group rather than the no dementia group. And so this really suggests that performance-based measures involving fine motor skill have the potential to identify dementia progression. And really what a take home from this work and that we're exploring more is that different motor tasks or different motor-based tests likely have varying levels of overlap with cognitive processes, which is likely how these different tests do or don't predict dementia onset. So where are we going from now? And I'm going to spend the last few minutes just highlighting some different directions that our group is going. And they really are taking us in one direction towards the leveraging animal models, and then the other direction is thinking about how we can implement some of our work into the real world. So building off of that Fulbright work that I just showed you, is it possible that these functional fine motor deficits may emerge earlier than cognitive deficits? And when we try and evaluate this in humans, this takes years to decades to assess. And the likelihood of individuals progressing all the way to dementia, even in biomarker positive individuals, is not very high. And so perhaps we can leverage animal models of Alzheimer's disease, particularly transgenic animal models that are genetically programmed to develop Alzheimer's-like pathology and progress to cognitive impairment in a reliable fashion in a matter of months. And so we've developed a rodent version of our performance-based test. This was published earlier this year in the Journal of Visual Experimentation. And then we can use some really canonical cognitive tests like the Morse-Water maze to evaluate memory or cognition. And what we have found so far is when we look at wild type versus transgenic models of Alzheimer's disease, the fine motor task shows differences between the wild type and the transgenic rats as early as 6 months, whereas the Morse-Water maze, sort of like purely cognitive measure, doesn't seem to be sensitive to changes until 12 months from now, or excuse me, 12 months of age. And so we are kind of expanding this to a longer longitudinal cohort and leveraging some advanced neuroimaging to allow us to longitudinally assess individual animals, and then validating that with some gold standard histology. Then looking to sort of real world applications, I want to talk about two of them. The first one relates to older adults experiencing homelessness. So we know that 1 in 5 adults experiencing homelessness is over the age 50. And nationwide, this is the fastest growing group of individuals experiencing homelessness. And kind of not surprisingly, the rate of dementia is twice that of housed older adults in this population. But going back to the very beginning of my talk, where I mentioned these existing measures in which that evaluate functional impairment. Well, how do you evaluate functional impairment in adults experiencing homelessness? For one, they likely don't have a reliable informant, and two, some of the predefined tasks may not be relevant for an individual experiencing homelessness. And so some examples of those are listed here. And so we have this idea that perhaps a performance-based measure that's quick and easy to administer can help us give insight into dementia or dementia proximity in unhoused individuals. And to do this, we've been able to leverage some infrastructure here in Phoenix. So the Haven is one of the only homeless shelters for seniors nationwide, and it is actually a renovated Super 8 motel in North Central Phoenix. And we did sort of a quick data blitz in September of 2025, where we just collected our assessment as well as the MOCA in individuals who were residing at the Haven. And I mean, for one, not surprisingly, the only 25% of individuals that we evaluated had MoCA scores above the 26-point cutoff. And this replicates some earlier work that my collaborator, Dr. Heather Ross, has published. But also we looked at what is the relationship between our assessment and the MoCA, as well as sex, age, and education. And as expected, based on our prior work in House individuals, there is a significant association with MoCA. And so it is tapping into these cognitive processes. But as we've also shown in other work, it was not associated with participant sex, age, or level of education. Then when we compare these sort of effect sizes and these effects to a housed population, we see the same association with the MoCA as in a housed cohort. So this is really sort of validating the generalized use for our assessment in a number of populations. And so there's a number of next steps that we hope to take with that moving forward. And then lastly, I just briefly want to touch on a collaboration with the very well-established and long-running Tsimane Health and Life History Project. And so the Tsimane are a group in sort of the Amazonian forest, rainforest in Bolivia. And this project that I have just been newly associated with through Margie Gatz and Ben Trumbull, that has been a long-running study that really is interested in looking at the prevalence of dementia and mild cognitive impairment in the Tsimane and other populations, because relative to sort of national prevalence and international prevalence, the Tsimane actually have very low rates of dementia and mild cognitive impairment. And so, but these individuals are a foraging horticulturist group. They have no education. They don't have a number system. There's not a lot of applicability of a lot of standardized cognitive assessments in this population. And so we have implemented our assessment in their longitudinal data collections. And actually, we sent like a plastic version of our test down to Bolivia. And one of the participants thought, you know what, I can do better. So he actually went home that day, crafted out of wood a version of our assessment. And that's now what's being used for data collection in this project. And at this time, there's been about 500 participants that have been collected, and they've said that they can collect about 6 to 8 individuals on our assessment per day on one of their data collection days. So there's a lot of exciting stuff that we hope to explore with this. And so to wrap up my talk today, I've shown you all this before, but in addition to that, we have data to really support the use of this assessment remotely and unsupervised. It can predict change over time, perhaps even longer, and really we're expanding our work to animal models, as well as implementation science, which is a goal of mine kind of in this next stage of my career. I want to do a shameless plug at the very end. If you know anybody that is interested in doing a PhD, or anyone on this call that might be interested in doing a PhD, please feel free to reach out to me after my talk today. So that's all I have. Thank you for your time, and I'm happy to answer any questions. I can't see who's on the call with the way that I have my screen set up, so please unmute yourself or put your question in a chat. Thank you.
Yeah, thanks so much. Great presentation. So I have one question. So in addition to education and sex, have you looked at mental health, like depression, anxiety? Usually those also play around with MoCA scores. I don't know if they also interfere with the motor, the performance of your test. Have you looked into those things?
We have not explored that effect, the effect of depression on our test in a kind of community-based sample that does not have strict inclusion or exclusion criteria on the GDS or DEDS, some sort of depressive scale. So in my clinical collaborator cohorts with Dr. Kevin Duff, who is now at Oregon Health and Science University, we have collected, done a quick depressive scale at every data collection. And those would be individuals who are cognitively unimpaired, amnestic MCI, or mild AD, as defined by the ADNI criteria. And we don't see a relationship with our test on I think it's the GDS that we use. However, there is a GDS cutoff to be in that study at baseline, but we do collect the GDS, at baseline and then at that 1 year follow-up. And so for individuals who, don't have, overt or very high levels of depressive symptoms, we don't see a relationship. So I don't know if that is a great answer to your question, but at least in the clinical cohorts that we have for whom we've collected GDS, we don't see a relationship with our assessment.
Yeah, thanks so much.
Yeah.
Okay, so everybody quiet. So anyway, so I have another question actually.
Okay.
So we work with Hispanic community here. So we're like, I'm, positioned in the valley. So we're just, next to the Mexican border. We have like 90% of the population Hispanic. So, so this test have been, studied into like large community of Hispanic, or just is still in the test, like the test phase is still not, I'm just asking because like how to implement this in a community setting. That's my question. If we want to.
Yeah. So something that I didn't mention that I should have in describing the MindCrowd cohort is the MindCrowd project partners with Target Latino, which is a marketing agency that specifically works with kind of minority populations. And so that's one of the ways that they've been able to enrich for Hispanic Latino participation in the MindCrowd cohort overall. And then also in the mobile lab, kind of going across the state, getting into communities that are probably similarly represented of the communities that you serve. And the MindCrowd cohort, when an individual is being asked if they want to participate, they can sign up to have all of the one, communication, and two, all of the assessments done in Spanish, including our assessments. So if somebody kind of clicks the box in MindCrowd that they want to do MindCrowd in Spanish, then we mail them a kit with instructions in Spanish, and the scoring side and everything is all in Spanish. That is not a very strong answer to your question, but that is a step that we've taken to to accommodate a larger and more diverse set of individuals in ways that I think not a lot of other studies can or have done. So I don't know what individual barriers you might need to overcome to implement something like this in your local community, but we've been able to show in some other work just sort of how easy the test is to administer if you had an experimenter or you wanted to do it face to face. It takes very little, basically no training to do, and it takes just about 3 to 4 minutes to administer. There's no sensor or kind of technology. There's no app or anything that you have to set up. And so I think a lot of the technological barriers to implementation, or at least scalable use across different platforms, are not applicable, I guess, in this case. And that is by design, because we wanted something that would be as simple and easy to implement. Um, so again, I don't know if that answers your question.
No, it does.
But
So I thought, so when you do the test, you do the timing, correct? Or, but I thought also but you don't have any sensors to measure the
not for
movement.
not for any of none of the work that I presented today
Okay.
involves any an instrumented version.
Okay.
My background in movement neuroscience, a lot of my colleagues and some of the reviewers for some of the work, they really want to see a lot of kind of motion capture. And so to kind of appease those reviewers, we can collect, aspects of the task, and we've published on those. But really, when you look at kinematic data, it correlates with time. And so, we can do deeper measures. Those don't seem to tell us anything that time above and beyond can't tell us.
Yeah, that's very helpful. Yeah, thanks so much.
Yep. Yep. I have no idea what time it is, so I don't know. My clock is covered by, so I don't know if I've gone over, but I'll just let you.
Yeah, I think there is no other question so far. Yeah, I think I don't see anybody asking questions. So yeah, thanks really for joining us, and a great talk, and thanks. Thank you.
Yeah, thank you. And my email address is shown there if you have any questions just about our work, or if you would like us to send some of the papers that I've that I talked about, definitely feel free to reach out. And thank you for your time.
Thanks. Thank you. Have a great day. Thanks.
Okay, thank you. Bye-bye.